morning, everybody. Bobby here, back with you. We're continuing with our studies on biology for the Neet and life sciences for the Net examinations. Today I'll be dealing with the subject of many enzymes that require cofactors for activity and continuation, which I just finished. The portion just finished was for enzymes that require cofactors for activity, right? And I will be completing the additional portions on that. So the next topic would be water-soluble vitamins function as coenzymes and continued portions, right? So stay tuned, right? I'll just catch you in a jiffy, right?
Right, now onto the subject proper. Enzymes that may transform energy from one entity to another. Right? In many biochemical reactions, the energy of the reactants is converted with high efficiency into a different form. For example, in photosynthesis, light energy is converted into chemical bond energy through an ion gradient. In mitochondria, the free energy contained in small molecules, derived from food is converted first into free energy of an ion gradient and then into a different currency, the free energy of adenosine triphosphate. Enzymes may then use the chemical bond energy of ATP in many ways. The enzyme myosin converts the energy of ATP into the mechanical energy of contracting muscles. Pumps in the membranes of the cells and organelles, which can be thought of as enzymes that move their substrates rather than chemically altering them, to create chemical and electrical gradients by using that energy of ATP to transport molecules and ions. The molecular mechanisms of these energy transducing enzymes are being unraveled. The free energy change provides information about the spontaneity, but not the rate of a reaction. Gibbs free energy, or a delta G1, measures the amount of energy available to do work, hence free energy involved in a reaction is defined in terms of both heat and entropy. Definition: delta G is equal to delta H minus T delta S. When delta G is equal to change in free energy, a measure of useful work, delta H is equal to change in heat content, delta S will be change in entropy, and T is equal to is the absolute temperature in degrees Kelvin. Example, in burning glucose, example paper, delta H is minus 673 kilocalories. Delta S will be minus 13 kilocalories. Delta G will be minus 686 kilocalories. The free energy of a reaction, delta G, tells us if the reaction can occur spontaneously. If a reaction can occur spontaneously only if delta G is negative, such reactions are said to be exergonic. A system is at equilibrium and no net change can take place if delta G is zero. A reaction cannot occur spontaneously if delta G is positive. An input of energy, an input of free energy is required to drive such a reaction. These reactions are termed endergonic. Two additional points need to be emphasized too. That is, the delta G of a reaction depends only on the free energy of the products, the final state minus the free energy of the reactants, the initial state. Delta G of a reaction is independent of the path or molecular mechanism of the transformation. The mechanism of a reaction has no effect on delta G. For example, the delta G for the oxidation of glucose to carbon dioxide and water It's the same whether it occurs by combustion in vitro or by a series of enzyme catalyzed steps in a cell. The delta G provides no information about the rate of a reaction. A negative delta G indicates that a reaction can occur spontaneously, but it does not signify whether it will proceed at a perceptible state. As will be discussed shortly, the rate of a reaction depends on the free energy of activation, delta G plus, which is largely unrelated to the delta G of the reaction. Enzymes alter only the reaction rate and not the reaction equilibrium, because enzymes are such superb catalysts, it is tempting to ascribe to them powers that they do not have. An enzyme cannot alter the laws of thermodynamics and consequently cannot alter the equilibrium of a chemical reaction. This inability means that an enzyme accelerates the forward and reverse reactions by precisely the same factor. Consider the interconversion of A and B. Suppose that in the absence of, a reversible reaction of 10 raised to minus 4 and minus 1 over 10 raised to minus 6 and 8 raised to minus 1. I figure that is G raised to minus 1, and G raised to minus 1. And K is equal to B by A, where Kf by R is equal to 10 raised to minus 4 by minus 6, which is equal to 100. All right? Now, at the enzyme, the forward rate constant Kf is, here it's forward rate constant F by reverse rate constant R. Is 10 raised to minus 4, S minus 1. Here it's S minus 1 and S minus 1. And the reverse rate constant, Kr, is 10 raised to minus 6, S minus 1. The equilibrium constant, K, is given by the ratio of these constants. The equilibrium concentration of B is 100 times that of A, whether or not the enzyme is present. However, it might take considerable time to approach this equilibrium without enzymes, whereas the equilibrium would be attained rapidly in the presence of a suitable enzyme. Enzymes accelerate the attainment of equilibrium, but do not shift the position. The equilibrium position is a function only of the free energy difference between reactants and products. And 3, enzymes accelerate reactions by facilitating the formation of the transition stage. The free energy difference between reactants and products accounts for the equilibrium of the reaction, but enzymes accelerate how quickly this equilibrium is attained. A chemical reaction of substrate S to form product B goes through a transition phase S plus that has a higher free energy than does S or P. So this is the diagram of the transition state, the substrate, which is uncatalyzed. Here, free energy is uncatalyzed, free energy is catalyzed. You go for a product where free energy for the reaction shows the difference at free energy. And that's the progress of the reaction. The double dagger denotes, you see this notation of S double dagger, that has a higher free energy. So the S double dagger denotes a thermodynamic transition state, a dagger up, a dagger down, right? The double dagger denotes a property of the thermodynamic transition stage. The transition, that is delta G, is equal to delta G1 minus delta GG. The transition stage is the most seldom occupied species along the reaction pathway because it is one the one with the highest free energy. The difference is free energy between the transition state and the substrate is called the Gibbs free energy of activation is simply the activation energy symbolized by delta G dagger. So enzymes accelerate reactions by decreasing delta G double dagger. The activation energy, the combination of substrate and enzyme, creates a new reaction pathway whose transition state of energy is lower than that of the reaction in the absence of enzyme. The lower activation energy means that more molecules have the required energy to reach the transition stage. Decreasing the activation barrier is analogous to lowering the height of a high jump bar. More athletes will be able to clear the bar. The essence of catalysis is specific binding of the transition stage. I repeat, decreasing the activating barrier is analogous to lowering the height of a high jump bar, so more athletes can clear. The essence of catalysis is specific binding of the transition state. Next will be the formation of an enzyme-substrate complex is the first step in enzymatic catalysis. Much of the catalytic power of enzymes comes from their bringing substrates together in favorable orientations to promote the formation of the transition stage. Right? States in enzymatic substrate ES complexes. The substrates are bound to a specific region of the enzyme called the active site. Most enzymes are highly selective in the substrates that they bind. Indeed, the catalytic specificity of enzyme depends in part on the specificity of a binding. Next would be the active sites of enzymes. That's F6. The active site of an enzyme is the region that binds the substrates and the cofactor of any. It also contains the residues that directly participate in the making and breaking of these bonds. These residues are called the catalytic groups. In essence, the interaction of the enzyme and the substrate at the active site promotes the formation of the transition state. The active site is a region of the enzyme that most directly lowers the alpha G double dagger of the reaction, which results, delta G double dagger of the reaction, which results in this rate enhancement characteristic of enzyme action. Although enzymes differ widely in structure, specificity, and mode of catalysis, a number of generalizations concerning their active sites can be stated. Next, we'll be talking about the active site, which is a three-dimensional cleft formed by groups that come from different parts of the amino acid sequence. Indeed, residues far apart in the sequence may interact more strongly than adjacent residues in the amino acid sequence. In lysozyme, an enzyme degrades the cell walls of some bacteria. The important groups in the active sites are contributed by residues numbered 35, 52, 62, 63, and 101, and 108 in the sequence of the 129 amino acids. The active site takes a relatively small part of the total volume of an enzyme. Most of the enzyme acid, the amino acid residues in an enzyme are not in contact with substrate, which raises the intriguing question of why enzymes are so big. Nearly all enzymes are made up of more than 100 amino acid residues, which gives them a mass greater than 10 kD and a diameter of more than 25 angstrom. The extra amino acids serve as scaffold to create a three-dimensional active site from amino acids that are far apart in the primary structure, are often sterically constrained from adopting the structural relations necessary to form the active site. In many proteins, the remaining amino acids also constitute regulatory sites of interaction with the proteins, or channels to bring the substrates to the active sites. Active sites are clefts or crevices. In all enzymes of known structure, substrate molecules are bound to a cleft or crevice. Water is usually excluded unless it is a reactant. The nonpolar character of much of this cleft enhances the binding of a substrate as well as catalysis. Nevertheless, the cleft may also contain polar residues in the nonpolar microenvironment of the active site. Certainty of these polar residues acquire special properties essential for substrate binding or catalysis. The internal positions of these poles residues are biologically crucial exceptions to the general rule that polar residues are exposed to water. Right? These polar residues, right, are biologically crucial exceptions to the general rule that the polar residues are exposed to water. Substrates are bound to enzymes by multiple weak attractions. ES complexes usually have equilibrium constants that range from 10 raised to 2 and to 10 raised to minus 8. Continuing there, and corresponding to free energies of interaction ranging from about minus 3 to minus 12 kilocalories, moles raised to minus 1, from minus 13 to minus 50 kilojoules, mole raised to minus 1. The non-covalent interactions in ES complexes are much weaker than covalent bonds, which have energies between minus 50 and minus 110 kilocalories, moles raised to minus 1. Between minus 210 and minus 460 kilojoules, mole raised to minus 1. Electrostatic interactions, hydrogen bonds, van der Waals forces, and hydrophobic interactions mediate reversible reactions of biomolecules. Van der Waals forces become significant in binding only when numerous substrate atoms simultaneously come close to many enzyme atoms. Hence, the enzyme and substrate should have complementary shape. The directional character of hydrogen bonds between enzyme and substrate often enforces a high degree of specificity, as seen in the RNA-degrading enzyme ribonuclease. The specificity of binding depends on the precisely defined arrangements of atoms in an active site. Because of the enzyme and the substrate interact by means of short-range forces that require close contact, the substrate must have a matching shape to fit into the site. Emil Fischer's analogy of the Lochen key expressed in 1890 has proved to be highly stimulating and fruitful. However, we know that enzymes are flexible and that the shapes of the active sites can be markedly modified by binding of substrate, as was postulated by Daniel E. Koshland Jr. in 1958. The active sites of some enzymes assume a shape that is complementary to that of the transition state only after the substrate is bound. This process of dynamic recognition is called induced fit. Right? Remember these points. The next portion would be the Michaelis-Mentin Model accounts for the kinetic properties of many enzymes. The primary function of enzymes is to enhance rates of reaction so that they are compatible with the needs of the organism. To understand how enzymes function, we need a kinetic description of the activity. For many enzymes, the rate of catalysis V0, which is defined as the number of moles of product formed per second, varies with the substrate concentration S. The rate of catalysis rises linearly as substrate concentration increases and then begins to level off and approach a maximum at higher substrate concentrations. That is the graph for that. Before we can actually interpret this graph, we need to understand how it is generated. Consider an enzyme that catalyzes the S to P by the following pathway: E plus S, reversible E to S, reversible E plus P. Michaelis-Mentin, I think that's the pronunciation of it, equation. V0 is equal to Vmax, S by S plus Km. The meaning of Km is evident from equation 23 when S is equal to Km, the V is equal to Vmax by 2. Thus Km is equal to the substrate concentration at which the reaction is half its maximum. Km is an important characteristic of an enzyme catalysis reaction and is significant for its biological function. Significance of Km and Vmax values. Michaelis constant Km and the maximal rate Vmax can be readily derived from rates of catalysis measured at a variety of substrate concentrations. If an enzyme operates according to the simplest scheme given in the equation below, the derivation of Km and Vm is most commonly achieved and the use of curve-fitting programs on a computer. Now, the Km values of enzymes range widely and it lies between 10 raised minus 1 and 10 raised minus 7. The Km value for an enzyme depends on the particular substrate and environmental conditions such as pH, temperature, and ionic strength. The Michaelis constant Km has two meanings. First, Km is the concentration of substrate of which the half the sites are filled. Thus, Km provides a measure of substrate concentration required for significant catalysis to occur. That should end the portions for today. Thank you. We'll see you tomorrow.